Michi, before we go to our breakout groups, there were a couple questions that popped up in the chat that I thought I would ask you right now. Somebody was asking about what, kind of in summary, what is it about the sulfide springs that is what drives the mollies evolution? Is it just the conditions of the water or the bacteria in the caves, or are there other factors there that you could maybe share?
Yeah, that's a great question. And that's exactly what we're trying to figure out. We have largely focused on the water chemistry in the past 10 years. So we now understand quite well, the physiology of these fish and how they're able to tolerate the toxic conditions. But when you saw those pictures of the springs, right, they look very different. Turns out there's no other fish in there. There's no fish predators in there. They eat really weird stuff. And so many things are different between a regular stream and the sulfide springs. And it turns out those other things, they're also important evolutionary drivers. So we've learned that the brain morphology is different of these fish because they behave differently. They have the modifications to their sensory systems, to the way they see. They have changes in their life history and how they reproduce. They eat different things. They have different kinds of teeth. They have a different intestinal tract morphology. So all of these things kind of layer on top of one another. And while we're not really understanding all of that quite yet, there's definitely clear evidence that it's not as simple as just the water chemistry. There's lots of stuff that goes into it.
Earlier in the talk, you showed those drawings of what made everything look like a bone in the skeletons of the fish in the gonopodium. And somebody was wondering if the gonopodiums are actually bone, or is that cartilage, or is that some other tissue that forms that organ?
Yeah, again, a great question. Yeah, so it turns out the fin rays in the fish, they do have bony elements in them. They're very fine, very thin bones, but there's calcification in there. And so, yes, the structure, I'm not sure, can you still see my screen?
Yes.
The structures that you can see here internally, these are solid bone, but also the fin rays here, these are kind of thickened and stiffened fin rays that have the same structure as well. So there's bony elements in here. There's a cartilage in here, and then there's a lot of connective tissue essentially that holds it all together.
And then just after that slide, you were talking a little bit about Lecithrophy versus Macrotrophy. I hope I didn't butcher that too much. But somebody was asking if they aligned with certain subgroups. When you were talking about the genetic tree, do they align with those, or do they kind of hit or miss?
Yes and no. Most livebearers are lecithotrophic, so they just retain their eggs and give birth to fully developed young. But Macrotrophy has evolved multiple times. So if you look at this phylogenetic tree, it's just not like they're all in one place. They occur all over the place. But Poeciliidae, for example, which is, I believe, the clades somewhere in here, you have quite a few species that are macrotrophic. And then in other lineages, like the least killifish, Heterandria formosa, that's really common in the hobby. That's also a macrotrophic species, but it's kind of, it's on its own, and its close relatives don't have macrotrophy. So sometimes you have a little cluster of species with that strategy. Sometimes you have single species, but they're really dotted all over the phylogeny. It's not just in one place or in one group.
And can both of those Lecithrophies and Macrotrophies, are they able to store sperm for multiple spawns, or does one need repeated fertilization?
Yes, the livebearers, as far as we know, can all store sperm. Although in our experience from the lab is if you don't have males around, your reproductive rates in the females go down really quickly. So yes, it's possible for females to give birth after a year or so without any males, but I think the reality is that even though they can store sperm, most sperm that's actually there to fertilize, or most eggs actually get fertilized by sperm that was received relatively recently. But yes, all live-bearing species can do that. And they actually store the sperm mostly right within the ovary where the eggs are, which is kind of a fun fact.
Then somebody asked, I'm going to rephrase this question, but is it even possible to take like sulfur fish and move them into a different environment and have them survive, or cave fish and bring them into another environment? Or are they so defined by their niche that they can't live anywhere or any way else?
That's a great question. So you can take them out, and actually in the lab we keep these fish in normal water just like all the other fish we keep as well. So they don't need these extreme conditions. But in nature, the same story is not quite that simple. It turns out these fish suck at surviving in regular streams, and this is not because it's too good for them there, but our main hypothesis is that they're just terrible at eating what's in regular streams and in competing with other fish. So in other words, there has been some costs as they adapted to the extreme conditions. And yes, they can deal with a toxic gas, but they cannot deal with their neighbor anymore. And so there's definitely these trade-offs. And we believe that's one of the main reasons why these species or these populations are splitting into different species, right? Because one cannot live in the toxic environment and dies when it goes out there, and the others who have adapted to the toxic environment, they can't really compete with all the other fish in the normal environment anymore, and as a consequence you have very little migration, and these species are slowly drifting away from each other. So this is a really excellent question, and I have a student that's thinking quite a lot about exactly that question, trying to figure out what's going on.
So speaking of living in different environments, I'm going to ask two more questions, and then I think we'll go to the breakout groups. So some of these questions are getting fairly specific, but you were talking about the gill sizes being bigger in sulfide fish, and this person is asking whether you've looked at the gill or skin structure in sulfide residents versus fish that don't live in sulfides. And other than size, which you already mentioned, are there other peculiar differences that you see in like the structure, skin structure, ultrastructure?
Yeah,
between the two groups?
Also a great question. We don't know much about the skin, but we have spent a lot of time looking at the gills of these fish. So yeah, they're larger. They have larger lamella. So overall, the surface area of these gills is much larger. And that's not just because of the sulfide. I didn't mention this, but the sulfide springs are also hypoxic because the sulfide actually reacts with the oxygen. So these habits are not only toxic, but there's super low levels of oxygen in there. So there's barely anything to breathe underwater. But we have looked at, we mostly use kind of molecular biology approaches to understand what's changing in the gills, because a lot of physiological processes happen in the gills of the fish. And if you look at kind of the physiological profile of the gills of different populations, they're very, very different. So it's definitely not just kind of the size of the gills that change, but the way that ion transport happens across gill membranes, the way sulfide is detoxified, which also happens in the gills. Yeah, all of those things are very, very different between sulfidic and non-sulfidic populations. So lots of changes.
And then the last question that I'm going to ask before we do the poll is kind of a combination. Somebody was asking if there's some kind of pressure, environmental pressure, whatever, that results in the development of macrotrophy, and then part of that is aquarium swordtails macrotrophic.
Great question. So swordtails are lecithrophic. All of the Sulfur's are, all the swordtails on the platys, none of those are macrotrophic. Why are some fish macrotrophic and others? Well, there's quite a debate about that in the science world right now. One of my former students and myself, we have worked on some analyses that indicate it might be related to food resources and competition. So we find macrotrophic species primarily in environments that have low food availability and high competition. So did I get this right? They primarily occur in areas where there's, low food, low competition. Whereas Lecithrophy happens in high food and lots of competition. Other research groups very much disagree with that finding, and they found evidence that it might actually be related to parent-offspring conflict. It might have to do with strategies that moms might compete to resources with their offspring, and that might induce some weird evolutionary dynamics that causes basically moms to withhold energy from their offspring and only give it when they need to. So we see macrotrophic as something good often, right? It's like, oh, mom keeps giving, right? But no, maybe it's the other way around. Maybe mom is withholding and is being stingy and only giving when she really needs to or she really can. And so, yeah, that's a hot topic in Livebearer research right now that is not completely resolved, I would say. We don't know. Lots of good ideas out there, but we really don't know.
Michi, I'll just speak from a lot of the comments in the chat. You've gotten a lot of great compliments on your talk, and thanks so much. And so if you stay put, Kurt's going to work his magic with the poll, and then we'll go into the breakout groups and be good. So thank you so much.
Sounds great. Thank you, everybody, for coming. And I'll step away for a second, just go bathroom, and I'll be right back.
What's your latest project, Michi?
We just expanded our greenhouse facility. So lots more tubs, lots more fish. We just had the last 4 come in yesterday or the day before yesterday. So
Are some of those segregated into high sulfur tubs?
No, we don't get to play with the sulfur in the lab. It kills my students too. So the university is not very happy when we play with the sulfur in the lab. So we do it on a very, very limited scale, but all of our sulfur fish are actually kept in normal water. It's just not feasible. The amount of sulfide you would need, it's also highly explosive as a gas. Doesn't help us either. And so there's just so many logistical issues associated with it that we're just not able to do that, unfortunately. I'd love to.
Those are.
You've heard of the mollies we've got up here in Banff?
Say that again. I couldn't understand you.
You've heard of the mollies we've got in Banff, Canada, haven't you?
Yes, they live in some sort of a hot spring, right?
Right. There's mollies and gambusia there.
Yeah.
It's pretty cool. Mike Helwig actually could tell you. We went and saw it.
Did you catch some? What kinds are they?
We can't catch them. They're actually protected.
No way. Canada protects invasive species? That's crazy.
Well, and gambusia are illegal in Alberta, but they're protected because they're in a national park.
Oh, I can see. Yeah.
Yeah.
Yeah, I can see that.
So Michi, just a very, very dumb question here. Does the high sulfide change the pH and the KH?
Somewhat, yes. So it's not the sulfide itself, but when it oxidizes, it becomes sulfuric acid.
Right.
But the thing is, these are this is all limestone, so the water is super hard and super well buffered. Okay. And so the average pH is somewhere between 7.8 and 8.4. So in the sulfide habitat, it goes down to 6.8, 6.4. Which is significantly lower, but like if you compare it, to the Rio Negro, it's not really acidic. So it's just because there's such high concentration.
Michi, do you have to feed those fish any type of special foods because of what they're used to eating the bacteria?
Yeah, that's our biggest nightmare. Yeah, so some populations do just fine, and others don't. So like the thermales I talked about, or the Poecilia sulfuraria, they're super hard to keep in the lab. We've never managed to keep them longer than for, 14 months or so, and then they're all gone. And I think a big part to the problem is their diet. And we've been playing around with various diets. In particular, we've been working with like enrichment of fatty acids because they seem to be really high in some of those bacterial mats. But it's a continuous challenge. I think the food is our biggest impediment right now to keeping all of these species in the aquarium successfully.
I'm going to jump in with a question that's been bugging me since you showed the slide of the map. There's a big gap in Amazonas. Are there no live bearers in the Amazon basin? That to me seemed very unusual.
There's very, very few. Yeah, if you looked at the map, there was a kind of a few dots scattered in the Amazon basin. Yeah, but there's almost no live bearers in the Amazon basin. That's correct. I think part of the reason is that live bearers don't do very well with competition. So they're kind of most successful in kind of marginal habitats, that dry up and where it gets hot or salty or with low oxygen. And I think, in the Amazon basin, you have just this phenomenal diversity of species, right? I remember visiting a few years ago, in one place we caught 280 species in one river, right? It's crazy. And I think, they're just not very successful in those types of environments. And if you look where you find live bearers in South America, that's where they're mostly coastal. They're mostly occurring in kind of those elevated salinity levels where they have pockets where they might be able to evade some of that competition. But it's a glaring hole. You saw that correctly.
Well, then just to follow up, why are they less successful as competitors? It seems to me that their reproductive strategy is very adept.
Yeah, but it's their reproductive strategy is really good when they can just, pump out a bunch of babies, and then those babies grow up in 6 weeks and pop out a bunch of babies, and that works really well. We call this R-selected.
Right.
When you are able to basically just explode your population within a matter of a few months, you have none, and then suddenly you have lots of them. And those species tend to do best in area or in environments where there's low competition, but there's suddenly like lots of great resources. Like a great example happened a few summers ago here in Kansas. We actually have Gambusia affinis here too. It's not native, but they occur all over here. And so we have this long-term research site called Kansa Prairie that has a beautiful stream. It's called Kings Creek flowing through it. And 2 summers ago, we hit a horrible drought, like a massive drought that's kind of coming up all year. And Kings Creek dried up, right, into these pools. And in a regular year, you have a hard time finding a Gambusia. But over the course of about 6 to 8 weeks, as the creek dried up, all that what's usually the native fish, they all started to wither away and die. And by the end, there was thousands of.
I've kind of kept that number of tanks because it's if I go much higher, I end up burning out with the water.
I was going to ask you why you limit yourself. It's either you limit yourself because of money, space, or energy, or your spouse.
It's pretty much all energy. If I
Sure.
I have some space down there, and the tanks aren't expensive, so I could, and I've been thinking of adding a few just because I'd like to raise a few more fry, but once you get them out there and then work gets busy and you start just the fish suffer.
Okay.
Randy, I got a question for you.
Yes, Kurt.
I got some emails saying that our maximum capacity was reached of 100. Do you have any idea if we're limited, even though we're paying for this service?
We used to be able, we went over 100 last month.
Yeah.
And I noticed we leveled at 100, and I wondered if that was the case. I think Zoom probably, and it used to be you can't have more than 2 people on for 40 minutes unless you buy the next level, but you guys on the board were able to do it. I think Zoom is starting to slowly, with so much people, they're applying rules. and we must have hit a limit, and we will need to discuss if we want to raise it to go over 100 or not. Because I wondered why we were hitting just 100.
Yeah,
we could have had 30, 40 more people who wanted to get in.
Yeah.
Yeah, unless you buy the webinar license, you're capped at 100, and then they tier it off of that.
Okay.
I
Okay,
thought it
we'll
used to be.
discuss it and
I thought it used to be
consider
100
this.
people presenting, but other people could visit and see. But apparently that's not the case, because we were over 100 last month.
Yeah, so they're
I'm
probably
almost sure.
they're being used
So
a lot. So
Well, I think Zoom is slowly clamping down on their rules. So I thought that. I sent Willie a note. I knew we were going to need to discuss that.
okay, well, we'll talk about it. In
Yeah,
2
but
weeks.
we're there's plenty of room for people to get in now, because I think we're like 80 some by
Yeah.
the time we went to breakout rooms.
Yep.
Okay, is that it?
Okay, I just thought I would bring it up because.
Do you want to share anything?
Me?
Yeah, you have the floor. How many tanks do you have?
Me?
Yes.
Oh, maybe 12, 15, something like that.
And what are the general sizes, just to give us an idea?
110 the biggest and 2.5 the smallest, I guess. So
No, then what you average around 20s, 30s, something like that, or what?
yeah, I suppose like a
20
well,
or
40 breeders. I have a half dozen
Okay.
of them. So
Okay. Well, you haven't been on, you've been bouncing around, so we never get a chance to talk about yourselves. So I wanted to give you that chance. You acquire any new species lately?
no, I've been was that benign neglect is what Milo was.
When's the last time you
say.
got a new species?
It was probably spring when we did the order, that last order.
Wow,
at,
in March.
oh,
That was
yeah.
the week that the pandemic was just breaking out. I remember we were
Yep.
standing around getting our stuff, and that was the last time I was hanging around with people like that.
Yeah, I've been so busy since then. It's just I'm thankful I'm busy,
Sure.
but it's just been nuts. I mean,
Okay. Well, I wanted to
So-
give you a chance to say a little bit about your fish keeping, because you're always running the
some
show.
of
and-
some of the neglect has bothered me, but some of the wild-caught discus aren't doing as good as they should have. So, but
Okay.
if you don't
All
have
right.
the time, that's your own fault. So
And you are recording the question and answer in the other session, right?
Yep.
Okay, just checking on that. I'm going to move on. Somebody else
Okay.
want the floor to your name, where you're from, what kind of fish keeping, or something about your fish room.
If you do encounter each other, do you recognize each other as mates? Do you like each other? If it comes to copulation, we know that gonopodia can be very different. Are the genitals compatible, right? So there's all of these layers that I think in nature, like in our case, you have the sulfur mollies here, you have the non-sulfur mollies over there. They're 10 yards apart, yet they don't reproduce with one another, right? And I think it's because you have these layering of barriers on top of each other. You take those barriers away, you bring them into the lab. Most of the barriers are gone, and they will make babies together really easily. There's a possibility that the hybrids might actually not be doing so hot, and that's what we're currently working on. But in your case, I've seen those fish in the markets, right? The feeder fish, and I mean a lot of them are just in a horrible condition, right? And one thing is with mollies and lots of Livebearers, once they're in a crappy condition, it's really hard to get them back and to reproduce. So once they're over that hump, you can baby them as much as you want, and you might get them to live for a year longer, but you're not going to get any babies out of them.
They had
And
to
that
be
might
dewormed, that's for sure.
say that again.
They had to be dewormed, that's for sure.
Yeah. But that practice of having live feeder fish all over the country, that's actually one of the biggest problems right now in terms of conservation in Mexico. You have mollies, swordtails, Poeciliidae, Pseudosaphophorus. You find these all over the country now, even where they're not supposed to be. And it turns out they're some of the most invasive species in parts of the country where they don't belong. And it's all due to the spread of these feeder fish.
Not only that, there is also, especially around Cuernavaca, they have had these fish farms And then you have these escapees, so to say, and they start.
Tilapia.
Yeah, so now you have this, I think they call it the Yucatan swordtail or whatever it is, which is quite orange. And well, they're debating now actually whether that is a hybrid between, let's say, a pet shop swordtail and actually a local swordtail.
Right.
Yeah.
Yeah, it's quite awful. Mexico's is changing fast and not to the better for the fish, that's for sure. So Plecostomus everywhere now. Places where you could go snorkel 15 years ago and you would see, I don't know, 12 species of cichlid breeding, you know, on 20 minutes snorkeling. All you see now is Plecostomus. Thousands of them. They're everywhere. They're destroying everything. And it's
our next question comes from Jessica. If you want to go ahead and ask your question, and then Laura, you'll be next.
Sure, thanks. Michi, you showed how when you were working in the cave that you had the respirator on, but are there other safety measures you have to take above and beyond what you normally would for field work when you're working with these sulfide springs?
Yeah, having a team and clear deadlines for everything, especially in the cave. So we go in as a team, we work on the clock. You're not in there longer than 60 minutes. If the team doesn't come out, then half of team, the other half is going in, and the other half is going to look for help. I mean, knock on wood, we've had no sulfite-related incidences in the past. But yeah, we have monitoring equipment that tells us what's in the air, because there's not only sulfite in there, there's also carbon monoxide, which is also not very good for you. And yeah, if you're in the wrong spot in the cave, you take your respirator off, you get tired very quickly, and that's just it. That poison is affected of the cave. And then some of the locals will go in with you, they refuse to wear a mask, and they're like, well, I can't even smell the sulfite anymore. And it's because the sulfite literally is paralyzing their smell receptors in the nose. You can't smell it because, you know, your cells are shutting down. And so, yeah, we have never had any issues, but we're really careful. But one of the springs, for example, is they built a swimming pool around it. So you can go swim in the sulfite spring. And of course, they chose the most toxic of all springs. There's like signs everywhere that it's dangerous. And of course, on Sunday, people come, they drink beer, they hang in the pool. And then people drown every now and then. It happens on a regular basis. And the people just say, well, they were too drunk to swim. Yeah, I don't know about that. I would not sit in a pool with my nose right above the water surface there for extended periods of time. And you get headaches, you get nauseous. The good thing in the surface springs is just that, you know, there's always a wind going, so it's not that bad there. But yeah, it's definitely, you know, it's nothing we take lightly. And it stinks really bad too. So I have a box of sulfide fieldwork clothes in the garage. They're not allowed in the house because you never get that smell out. And your skin smells even weeks after you take a hot shower and you're like, where is this whiff coming from? I think it's deeply embedded in your pores.
Thank you.
Sure. Who's next?
Laura, if you want to go ahead, that'd be great.
So my question was, so you touched on this a little bit with the cave fish, but I was wondering about the ones that are on the surface sulfur springs. How far divergent are they from these more clean freshwater streams?
Yeah, so it turns out that's really different depending on what spring you look at. So early on, we mostly used like mitochondrial DNA to estimate that. And when you do that, it looks like they're super old, but it turns out That's just because there's some really weird things that happen evolutionarily speaking. So what we've done since, we've sequenced the genomes of all of these. And what's really cool is when you do that, you can use the DNA to make estimates about how old or how long ago that divergence was, right? And so in the oldest lineages, it's about 8 to 12,000 years. In the youngest lineages, it's between 500 and 800 years. So there's some of these weird looking sulfide springfish. If you would have arranged it just right and you came over with Christopher Columbus and came for nothing but seeing sulfide mollies evolve, you could have watched them all along the way. So yeah, it's definitely not something that took hundreds of thousands of years. Selection is really, really strong in these environments. And whenever selection is strong, evolution can happen really, really fast. And I think that's what exactly has happened here.
Interesting.
Did that answer your question?
Our next question comes from Allison. Go ahead, Allison.
Yeah, so I was wondering if you looked at the behavior differences between the sulfide fish and like close relatives. You know, you talked about, for example, that throughout these poeciliids that the aggression levels can change. Have you looked at like things like aggression level or other behaviors?
Yes, we have. So I have not done any of the aggression work, but a colleague from Germany has, and they found that the sulfur springfish, they are all a lot less aggressive compared to the non-sulfur fish. And the interpretation of that is mostly, like, sulfide fish don't do anything that costs energy, because they don't have any. You know, they have to breathe at the water surface. They don't have time to eat. Detoxifying sulfide is metabolically really expensive. They have really inefficient mitochondria and energy production. So the idea is they kind of shut down all the superfluous stuff like aggression and don't engage in it that much. What we have done a bunch of work on mate choice.
Mm-hmm.
And the sulfide springfish, they're not very choosy. They don't have very strong preferences, but it turns out the non-sulfidic fish, they have pretty strong preferences against sulfide fish, which makes sense, right? It's unlikely that a non-sulfur fish goes into the sulfide spring. It will just die because it's not adapted. But the sulfur fish, they can hang on in the non-sulfidic environment. And so there's some evidence for, I'm going to use a technical term here since you're Mike's grad student, but some evidence for reinforcement, essentially, around those boundaries, habitat boundaries.
And have you looked at like reproductive levels? Are they similar reproductive levels in the sulfide?
No, very different life history strategy. So your regular, more, you know, Atlantic molly, Poecilia mexicana will make, depending on the size, anywhere between 20 and like 120 babies per clutch. Sulfide spring mollies, they will make like 1 to 4, but they're by mass about 5 to 10 times larger. So they make these large, giant babies. And that can also be explained simply by energy limitation. So in environments where energy is scarce, fish tend to make large babies. You basically give them a snack pack along the way, right? Whereas in an environment where energy is abundant in the environment, you just make as many babies as possible because they'll be fine right on their own. And so yeah, that's another thing that consistently changes. No matter what sulfide spring you go to, they all make giant babies. Another great examples of convergent evolution. And it stays in the lab. That's why it sometimes sucks to work with those species, because, you know, you baby them and baby them and you baby them, and then you get 4 babies. We have one line of Poecilia sulfuraria that we've had now for almost 2 years, and we have one female that drops, and she makes 1 baby at a time. We've never gotten more than 1 baby, and it's not because she eats them. She doesn't. She just makes 1 baby at a time. Well, we've gotten 3 recently, so.
Eric, did you want to go ahead and ask your question?
Yeah, sure. Can you hear me?
Yes.
Yeah, we can.
Thank you, Michi. I've noticed with the Livebearers that I keep, you know, kind of the basics, that the male's attempts to copulate with the females is really heightened as they get close to giving birth. And I've often wondered if those attempts are somehow needed or used to actually trigger the start of the birthing process.
I don't think so, but your observation is absolutely spot on. When you have a female that's about to give birth or just gave birth, all the males in the tanks go crazy, right? And the reason for this is this. So they can store sperm, right? Poeciliids can. But the thing is that usually the most recent sperm is actually the one that's successful in fertilizing the next batch. And it turns out that the females are receptive for that next batch of eggs, right? To fertilize that next batch of egg right before and after a clutch is born. So that's really when you want to make your investment, right, in terms of depositing sperm. Because if you can catch a female right around when she's giving birth, she will have to have another batch of eggs ready to be fertilized. All your copulation attempts in between, you know, where will the sperm go? Well, it might be stored, and if you get lucky, you know, you might be able to have a fertilization, but it's really because that's really the critical time for the fertilization process. And I think it's more that the birthing and that the you know, the birth about to happen or just having happened, that's really a signal to the males that, you know, now is the time where you can kind of maximize your reproductive success rather than you know, the males helping the female. I think the females are really kind of the victims in this play, and it's not that they like need that stimulus to give birth, if that makes any sense.
Do you think the males are triggered by sight, or is that somehow a scent thing?
So I don't know about for all poeciliids, but in the mollies, I bet it's by scent. The reason I'm saying that is that mollies and many other life bearers too, they have a behavior that occurs just prior to copulation. It's called nipping. I'm sure if you've had life bearers, you saw this before, right? The males would kind of follow the females and they will kind of nip at the gonopore. And it's not, you know, well studied what that behavior does, but I think it's really plausible to assume that there's some exchange or some sensing of chemicals or pheromones, because, you know, the skin on the head is covered in taste receptors in these fish. So they don't only have taste receptors in their mouths, they have them covered all over the body. And they kind of close physical contacts. I would not be surprised if the males are able to detect kind of a hormonal change or something like that in the females that makes them able to tell whether, you know, birth is imminent or not. Like we know in swordtails, for example, and that this is the case. It goes the other way around there. There it's actually males that release chemicals in their urine during courtship, before they're mating. And females are essentially tasting or sensing that urine because it has species-specific cues in there. So the males are actually able to tell, the females are actually able to tell, is this a male of my species or a different species, by getting those chemical signals during the courtship, during mating. And when you have pollution and those signals get interfered, you get hybridization in nature between different swordtail species. So we know
Thank you very much.
Communication is important.
Thank you so much.
Sure.
Kurt, we had a request from Laura to be moved to the fish room. If you could shift her over, that'd be great. And then Eleonora, you had your hand up. Do you want to go ahead and ask your question?
Yes, thank you. First of all, talking about gonopodium, I have seen how a female heterandra basically took a male out of business by, she was so bugged by him that she bit off his gonopodium. So he was really, you know, he was out of business for a few weeks. That was, I mean, I thought it was really a nice thing to say, but it doesn't have anything to do with my question. There are some Mexicanas that have a very nice yellow seam on the tail.
Yes.
It's beautiful. But Do you know, it seems that it's not a dominant trait because I have noticed I left some of those mollies that I caught in Manialtepec in actually a sulfurous hot spring in close to Puerto Escondido with the biologist that I know, and they got mixed up. And in the next generations, the yellow seam disappeared. It became like a regular Mexicana.
Yep.
So do you know whether that is dominant or not, or how that basically genetically reproduces?
It's not genetic, yes. So in many life bearers, you have these genetic color traits, right? Like in the platy fish, you know, the tail spotting pattern is purely genetic. In some of the swordtail species, Mike Ryan is one of the reasons why Mike is famous, right? He discovered that there's like a simple genetic basis to different male morphs. But in the mollies, that's a little bit different. There's maybe a genetic component to it, but I think it's largely environmentally driven. So in all populations in nature, you get amazing males, right? The ones that you just described that are getting huge, they're yellow and blue all over. But then you also get these puny little things, right? That are basically swimming gonopodia. There's not much more to them. And they're gray and boring. And you know, you can take the most beautiful males home, and then in the next generations, they're all the puny little fish. And part of this is a matter of space and density. So that's what we see in our fish room. If you keep them in small tanks, you will get puny males. If you keep them in high densities, you will get puny males. So if you really want to get those nice males, you have to give these fish space, and you need to make sure that you don't keep them in too high densities. We in 10-gallon tanks, we get some pretty colorful males, but they're all fairly small still. In 40-gallon males, we have about a dozen 40-gallon tanks, and I would say in 2 or 3, there's a really nice male in there. We're talking, you know, 3 to 4 inches, perfectly yellow and blue, the way you would pull them out in Mexico. Our 150-gallon tubs in the greenhouse, you get nice males, right? There's a nice male in almost every tub. And so, yeah, I think if you really want to, and good food is also like lots of food. We feed our fish like crazy. And unless you really give, you know, like almost feed them to the death, I think those are some of the critical ingredients you need. And so, yeah, I think it's all about maintenance condition in the mollies. And I think it's similar for sailfins too. I don't know. Allison, you guys have more experience with those. I haven't kept sailfins in a long time. But it always seems like you can get some nice males, but then in a fish tank, most of them mature really early and don't get very pretty.
Thank you.
If I don't see any other hands up, Willie, you had a question about raising your hand. Did you want to go ahead and ask Michi a question? And then I think the way the pace is going, it doesn't seem like we're going to have so many questions that we'll be talking over each other. I think after Willie asks this question or whatever, you can just feel free to unmute on your own, and let's just be respectful to each other. If somebody's starting at the same time, maybe defer to them. So
okay, great, thanks. So Michi, one of the slides that you went through rather quickly was the spread, or really across the Americas. Okay, and I noticed also the offshore islands. You know, I think it looks like Cuba and so forth, also had those. So, and then you talked about rapid evolution and adaptation to sulfur springs. So you seem to have an example of very dramatic very long-term old divergence versus new ones. What kind of differences do you see in the diversity of these species?
So, I mean, we're studying the sulfide springs mostly because they're convenient, right? Because there's strong selection, and we kind of can tie it all back to some very, very specific things. And sometimes it's really hard to kind of understand, you know, why you have one high diversity in one place versus, you know, low diversity in other place. One of my colleagues, he studies fish mostly on the Caribbean islands, and he's always just puzzled by the fact that, you know, I don't know, there's like 20 some, almost 30 species of Limia on Hispaniola, and there's one on Cuba. Why is that, right?
Yeah.
And I always say, well, it's because there's Gerardinas, so there's other fish that already, you know, filled those niches maybe, right? But I mean, who knows, right? Like, we don't really know. And in many of those older lineages, we just don't necessarily know, you know, how the species originally originated. We don't have a time machine that allows us to go back to that splitting event, right? And that makes learning about speciation really difficult sometimes. And that's where we're lucky in the sulfide springs. It's like happening right now. It's in progress, right? Most of these things are not two species yet, but they're clearly also not just one and the same thing. And so it allows us really to understand, like, well, how does this unfold, right? And when you look at some of those deeper splits across islands and across different geographic regions, you have to, you know, rely on different methods and make much more indirect inferences, which, you know, it's just not the way I learn about nature. It's like a great way to learn about nature, but it's not my specialty. And I'm definitely, you know, holding out for the time machine so I can go back and actually see how it happened. But there's, you know, some brilliant scientists out there that use different methods to infer some of those speciation patterns at deeper scales.
Okay, I get it. Thanks. So your perspective is actually, if you think about it, you're really specializing on some on some niches, right?
Yeah, very much
okay.
so. Yeah,
all right, I
like,
get it.
You know, in real life, I kind of work through life like this. But it's always this trade-off, right, between being able to dig in and try to figure out something very specific and not losing track of the bigger picture. Because if you think about it, I mean, who cares about these little fish in sulfide springs, right? I mean, like, if I think about all the things that are going wrong with our world, like, why on earth would I devote my life to little gray fish that live in nasty springs? But you know, the truth is that you can learn a lot about general principles, right, that govern how diversity came about and how nature works by digging in really deep into those individuals or very kind of random systems. And you know, that's how we learn about things. If you always just sit at the big picture, I think you're missing out on a lot of the details that brings you forward. And I think this is why the scientists have the tendency to dig in. You cannot, you know, if you think about the circle of knowledge, right, you cannot push the boundary all around the circle. All that you can do is kind of bop the boundary and expand our knowledge in a very little specific area. And that's why we sometimes might seem a little obsessed. Well, maybe we are a little obsessed sometimes. You gotta be.
Yeah.
Does anybody else have something they'd like to ask? Willie, as president, do you have any wrap-up remarks then, or?
Wow. Actually, you know, it's sort of with Michi, it's like, how many areas do we want to go? Because you worry that sometimes, you know, there's several threads that, well, is that just going to be a little too ditchy and too geeky, or just you know, our personal interest? So I do want to go back. I would just say one thing, Michi. I think, so, you know, for most of us, not everybody here, apparently I'm just very impressed by this group, but for many of us, you know, our only exposure are the little ornamental fish that we have, you know, in our tanks, right? And these are the fish that we all started with, right? If you had a tank, if you had a 10-gallon tank, right, you bought these fish to start off with, right? And you killed them horribly and so forth. And then, and then some of us after 20, 30 years in the hobby have sort of rediscovered them, either the wild species or wild type of fish. So from an aquarium perspective, what do you see when you see other people's aquariums where they're keeping live bearers that they're doing wrong?
Oh man, that's a hard question. Most people I interact with, they love live bearers, and they're mostly much better fish keepers than I am. I think the first thing you can do wrong when you want to keep live bearers is go to pet store and buy some. That's the biggest mistake, because invariably your pet store live bearers are in atrocious condition. All the mollies are what Eleanor described, right? She brought them from the market, and they never did well from them. And if you look at most of those fish, they're over the hump. You can take as good care of them as you want. You will not get them to reproduce. You might as well throw your money out. So, get your live bearers from a club member. That's like the first important rule, because those fish will do well for you. I have had some surprising good luck on Aquabit, I have to say. There's some really good sellers on there, but your club is where you should get your live bearers from, because you will not have fun with the pet store live bearers. You won't. They will die on you, and you will keep buying them, and then you might have some babies, but they're just, they're bred by the millions in Southeastern Asia on farm or in Florida. Those fish are mass produced. They're not healthy. They're not worth your time. You're not going to have fun with them. But otherwise, most of the basic live bearers are pretty easy to keep. Change the water and have good filtration. It's all about the water. Fish keeping is all about taking care of water, right? That's always been my philosophy. And then secondly, it's about food. And depending on the food you give, it becomes water again, right? Because all the waste ends up in the water. And so what I think about when I think about fish keeping is all about, what do I feed my fish, and how do I make sure that what I feed them doesn't kill the water? Because what's good for the fish is bad for the water. And I think that's the challenge, right? And I think if you get into some of the more finicky live bearers, species that are small, species that have more strict dietary preferences, then you really need to think about food, because you're not going to get by with just commercially available flake food and granules like that. You're not just going to get by on brine shrimp. So live food culturing, I think, is something that really can lead you to some breakthroughs with some hard to take care of species. Also in Europe, where I grew up, we all go out and catch our fish food. It's called ponding, loosely translated. Like every serious fish keeper has a dedicated bucket and net, and all year long we go out to like the city pond or wherever, and we go out and catch plankton and feed our fish live food from the ponds. And then everybody's like, oh, think of all the diseases that the fish can get. It's like, well, think of all the nutrients your fish can get, right? And so, like when I was a kid, I had a separate fridge in the garage just to like have my live Daphne that I would catch every Saturday morning, right? That's what a lot of us feed the fish. And you guys up north, you have tons of ponds around. Just get yourself a net, and it's amazing what you can suddenly breed that never bred for you if you just give them a mostly live diet. Of food items that we're eating on algae and all this stuff in the pond, and bringing back all those nutrients, because when it comes down to it, your fish food that you buy is a complete diet. That's what it says on there, right? It has all the protein, lipids, the carbohydrates, and the vitamins, and the minerals that your fish need for survival. Not necessarily for thriving, right? And I think, like when you're serious about fish, like for me, being serious about fish keeping is all thinking about, yeah, what do I feed them? How can I keep the water clean? That's really the challenge. And I think that extends to many other species too.
Michi, we had a question from Jim. He was asking, what are one of your top 1, 2, or 3 species that you'd like to see make it into the semi-mainstream aquarium hobby? You mentioned a couple of them at different points in your talk, but is there a recap of like your 3 stars you'd like to see getting into the hobby better?
I really would love to see the Poeciliidae in the hobby more. They're gorgeous and they're quite imperiled in nature, all of them. You know, like it's not something I would like to see like in a pet store, but I would love to see a group of, you know, dedicated hobbyists really taking care of that. There's also some of the northern swordtails, which are becoming, you know, rare and rare in nature, like Sulfur malinche. If you go on the website, I think it's CARES, it's this you know, group of aquarists that tries to keep fish around for, you know, kind of as a backup essentially to the natural populations. I'd love to see that. I would love to see more hobbyists maybe just dedicating a tank or 2 to keeping some fish that have a rough time in nature. But you know, like we literally don't know, right, what's gonna stick around. And I'm not sure whether you guys have ever heard of James Langhammer, but he's a fellow of the American Livebearer Association. And back in the 70s, he got some livebearers of different family, good aids, for Robert Rush Miller, who was a scientist at the University of Michigan State, one of those 2, some university in Michigan back in the day. And so these goodaids, these are fish that, you know, they live in desert springs in kind of north central Mexico, highly imperiled. And Langhammer has managed to keep several populations alive for over 40 years in these tanks. Meanwhile, some of these populations disappeared in nature, right? That was one guy. And he's getting up there in age, and I think he reduced or almost entirely quit his fish operation. But like, you know, I think that's something where, you know, you can get some of the joy of taking care of your hobby and kind of mix it with some greater purpose too. And there's a variety of livebearers that will kind of qualify for an endeavor like that. Also, Alpharo cultratus, the knife livebearer, that's just an amazing species. If you have them in the right light with some floating plants, and then they have these awesome courtship display. The males will swim over the females and flicker their ventral fins over their eyes. Gorgeous. I don't know why anybody would not have that in their fish room. But the answer is all of them. All of them.
You know, I wanted to ask a question that may be just folklore or whatever, but it might speak to the condition and the ability to bring fish back. One of the things I heard somewhere along the way is that when fish condition begin to decline, one of the first organ groups that begin to suffer major degradation are the reproductive organs. And even so, if you get a fish and you manage to get it looking fat and healthy, you may never get the fish to bring that part of its body back to, you know, prime condition. Is that true or not true, or does that have any basis in fact?
You know, I honestly don't know. And I don't know whether anyone has really studied that rigorously. From observation with livebearers, I would say it's totally true. If you get your fish in a bad shape, there's no saving grace. You might be able to fatten back up, but most often that's not even the case. And if you think about it, you know, from an evolutionary perspective, they kind of make sense, right? These are fish species that in nature, they probably rarely get a year old, right? They're born, they grow to maturation in like 6 or 8 weeks, they reproduce, live fast and die young. And so essentially, those species have evolved to really invest most of their energy into reproduction, because they're not going to know whether they live the next day. So why would you take care of your body if you might get eaten by a predator anyways, right? And so we notice, for example, from annual killifish, right? You might have heard of these killifish species in Africa and South America. They live on the Cerrado in South America and in the savannas of Africa, in places that have really seasonal rainfall, right? And so, it starts to rain. The killifish eggs are actually dormant in the ground, in the soil, right? Water hits them. They start developing. They're adults within 2 to 3 weeks and spawn, and they die, right? And you bring those fish into the laboratory and you give them the best and happiest condition, and they still die after a few months. And the reason for that is, right, in nature, like, these fish never live more than 6 or 8 weeks. So imagine they had any kind of a mutation in their genome that causes them to have cancer at week 12. Who cares? They're already dead then anyways. So all of these mutations can stick on. Selection will never remove some of those mutations. Mutations that make them live longer are completely superfluous, because they're going to die anyway, because there's no water, right? And so a lot of these small-bodied species, I think they just don't have the evolutionary history to where they were selected to invest into repair and maintenance, right? And so, yeah, once they're over the hump, they're kind of done. And I bet you could find, examples in longer-lived species that are the opposite, right? Where you can really nurse them back from the brink of death, and the next thing you know, they're spawning for you. Isn't that how cyclic keeping works? Right? They all beat each other up to near death, and then next thing you know, they're having babies. But yeah, I don't know. I don't think it's researched well, but kind of observationally, I totally think it's true. It's once you treated them badly, they will never come back and do good on you, the Livebearers. I think that's very true. And we have dissected many old females, right, or many large females that we know have been sticking around and have not been dropping fry forever. And you open them up, they're just full of fat. Their ovaries are teeny tiny, and they're just chock full of fat. So they look like they're doing great, but they're definitely not reproducing. But yeah, why? I'm not sure. I'm not sure anyone has a good answer on that.
If anybody else has a question, just feel free to unmute and go ahead and ask it. Otherwise, I'll turn it over to you, Willie.
Well, it's 9:30, so we are getting close to our time. So there might be just exhaustion here.
I'm still trying to get transferred to the plant breakout room.
They have snacks. They'll be going strong.
Yeah, right.
Going on. I'm trying to get transferred.
So Michi, so there's several firsts here. I think just the nature of the questions and just the number of people that I think logged in, not necessarily to participate in an MAS meeting, but to hear from Michi Tobler. So lots of names that I've met over the last 30 years, but I hadn't seen recently. So thank you very, very much.
Well, thanks for having me. I had fun. So
yeah, and I hope Allison, you can get an invite out of this. Okay.
Yeah, Allison, did you see the online forum we're having in February?
I did see it. Yes.
Okay, I hope to meet you there formally. Well, yeah, online again, I guess.
Yeah.
Okay, everybody. Well, thank you very much, and we hope to see most of you, if not all of you, next time. Our next speaker is Bob Kraus on fancy guppies. Okay, another Poeciliid.
More Livebearers.
Yes. All right. Thanks, everybody.
Nice to meet you all. Thank you.
Thank you.
Bye.